where the cells proliferate.
Cell cycle,
Mitosis and interface.
cell cycle,
What we'll be learning today.
cell cycle,
Cell cycle,
cell cycle,
cell cycle,
cell cycle.
cell cycle. Once upon a time, there was a lone cell who was responsible for holding the hereditary material of the body and performing many other important functions. However, despite how capable it was at performing these tasks, the cell realized that it needed more help in order for the growth, replenishment, and reproduction of the entire living body. So, using all of its cell power, the cell divided itself, resulting in multiple daughter cells that would further aid the body for the better. Our mother cell did this by utilizing what we call the cell cycle. What is that? We define the cell cycle as a series of carefully orchestrated steps by which cells make copies of themselves. Seems pretty simple, but this process is actually much more vast and complicated, so let's dive deeper. The cell cycle begins with interphase. This is where the duplication of the cell's contents occur. It is composed of the G1 phase, the S phase, and the G2 phase. It all begins with the gap 1 phase, also known as G1. In this phase, 4 things happen. The cell increases in size, cellular components except the chromosomes are duplicated, cell accumulates the building blocks and proteins needed for chromosomal replication, and the cell produces energy reserves for replicating chromosomes. It's then followed by the synthesis phase, or the S phase, which has 2 important events occurring. DNA replication occurs and the cell growth continues. The last phase is called the gap 2 phase, or G2, where the cell continues to grow. Cell replenishes its energy stores and synthesizes proteins necessary for chromosome manipulation. Some organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the M phase. I don't know if you guys noticed, but each phase and interphase always has the cell increasing in size. And try to think of the G1 phase as the initiation phase, where it first prepares anything necessary for the next coming steps, and G2 as the form of replenishing these materials used in G1 for the next round of the cycle. We should also mention that the cell can enter a phase called G0. Here the cell isn't dividing nor preparing to divide. The G0 phase acts as a resting place for the cell. When inactive, cells enter the G0 phase after they exit the cell cycle when they are not preparing to divide. In fact, some cells stay in the G0 phase permanently. Cells are signaled to move from G0 to G1 when it is activated through the process where the growth factor binds with the growth factor receptor in order to achieve RAS, a proto-oncogene. RAS would then send a signal to the DNA, causing it to express proteins for cell growth and division. This is the time the cell enters G1. Interphase is then followed by what we call the mitotic phase, where spatial segregation of duplicated contents occurs. Remember the acronym of PMAT? This represents the 5 stages of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, chromatin condenses tightly into discrete chromosomes. Let's note that each duplicated chromosome consists of 2 sister chromatids held together at a centromere. After that, the nuclear envelope and nucleolus begin to disintegrate, and outside the nucleus, the spindle fibers begin to assemble between separating centrosomes. This marks the end of the prophase and the beginning of the prometaphase. Observe that prometaphase follows the events of prophase as chromosomes continue to condense, and the nuclear envelope completely disappears. Spindle fibers from each pole begin to attach to the kinetochore of either side of the centromere. This is followed by the metaphase, where centrosomes are now positioned at opposite poles of the cell. Chromosomes then align midway between the spindle poles. The spindle fibers are now on either side of the chromosome and extend opposite poles to the spindle. Finally, the unattached spindle fibers reach beyond the equator and overlap. Next is the anaphase, where sister chromatids separate and become independent chromosomes. The spindle fibers start to shorten, causing these 2 independent chromosomes to begin moving towards opposite poles of the cell. The mitosis phase ends with telophase, where everything starts to return back to how it was when the cycle started. Chromosomes uncoil back into chromatin, nuclear envelope and nucleolus begin to reassemble, and spindle fibers disintegrate. And that marks the end of mitosis. The whole process ends with cytokinesis, which is the physical division of the cell into 2 daughter cells. Always remember that this is the division of the cell's cytoplasm. Here, cytoplasm divides by the invagination of the cell membrane, but that's only in animal cells. In plant cells, it's by the formation of a cell plate which will structure over time, but no matter what, it will always result in two daughter cells. What phase in mitosis is this? I think it's the metaphase. What phase in mitosis is this? I think it's the anaphase. You may be wondering, why is this all important? I mean, yes, they're cells, the building blocks of life. But why do cells need to divide? There are three main reasons we could tell you. Remember the acronym GRR. This stands for growth, replenishment, and reproduction. As you may have noticed, the whole entire cell cycle has a very specific set of instructions and steps to follow. It's very important that no mistakes are made. So, how does the cell make sure that no errors are made along the way? Well, the cells have specific regulation cycles using checkpoints to help with that. There are 4 checkpoints: the G1 checkpoint, the intra-S checkpoint, the G2 checkpoint, and the M checkpoint. If you notice this regarding the M checkpoint, they always check for DNA damage, which reminds us how important it is that our DNA remains free of mutations. In cell cycle regulation, there are 2 types of genes, the proto-oncogenes and the tumor suppressor genes. The proto-oncogenes encode for proteins that positively regulate the cell cycle, essentially promoting cell division. The tumor suppressor genes, on the other hand, encode for proteins that negatively regulate the cell cycle, basically inhibiting cell division. The G1 checkpoint going to the S phase is the most important, so let's look further into that. If there is no DNA damage, the cyclin E will bind with CDK2. This would cause RB to split from E to F. The E to F sends signals to the DNA, and DNA synthesizes proteins for DNA replication. This prepares the cell to enter the S phase. But if there is DNA damage, P53, the tumor suppressor gene, is activated. That signals the DNA to express P21, another tumor suppressor gene, which inhibits cyclin E from binding with CDK2. P53 pushes for DNA repair, and if the damage is irreparable, the cell is signaled to go through apoptosis, which is the cell programming its death. Well, that was quite a lot about cell proliferation. We hope you remembered all that. Let's test what you've learned with a short reviewer to conclude. In interphase, what are the 3 phases? G1 phase, S phase, and G2 phase. What are the 5 phases of mitosis? Prophase, prometaphase, metaphase, anaphase, and telophase. What is the process where the cell divides by the invagination of the cell membrane? This process is called cytokinesis. What is the most important question being asked during a cell regulation checkpoint? The answer is checking for DNA damage. That's all from us! Hopefully we helped make this topic a little more fun and easier for you. Till next time!